This is Kieran Barber's master's thesis presentation at Uppsala University in 2021. And he will talk to us about random graph models of a neocortical column in a rat's brain and their topological statistical distributions.
Hi. Yeah, that's what it is. My name is Kieran, and this is my presentation on random graph models of a neocortical column in a rat's brain and their topological statistical distributions. Cool. Okay. So we have three sections we want to look at. The first section is to just quickly talk about the Bluebeam projects and who they are, and also to discuss briefly on the brain. The second section we want to look at the models that we use to model connectome, which we'll discuss later. And the third section, we want to look at some results to compare these models to the connectome. So the Bluebeam projects are a Swiss research group, and their goal is to build a realistic model of the human brain. A couple of the reasons for doing so is to be able to stop animal testing and to address issues of neurological disorders. And so to begin this process, they had a proof of concept where they modeled some microconnectome in the rat's brain. In particular, they looked at the somatosensory region. And there's a view to scale this up to the human brain at some point. Okay. We want to discuss the development of the brain. This is taking sort of like billions of years to develop. One of the key developments was the neocortex that's needed to be able to sort of cope with living on land and used for like higher complex cognitive functions. As this cortex was forming, it became larger and larger with these columns, because these columns were considered like some sort of like final product. And so because it was considered so good, it just replicated itself. And so the brain was expanding in our skulls. Like we started to get like this sort of smooth surface, but because this replication just kept continuing, we needed to somehow pack more in. So to be able to accommodate this, the brain started to like fold in on itself, and so we get this image on the right hand side where we have like all these like grooves in our brain today. Yeah. So if we take a look at the brain and then we want to like zoom in on the surface, we have this sort of like the sort of surface view of all these cortical columns, and human brain has about 1 million of these. So if we were to take out like this column here, it's highlighted in the yellow, we'd get like this sort of general structure, like on the right hand side, where we can see it's split into several layers. Yeah. So because we were looking at a rat, so we also want to make a note that the rat has many of these neocortical columns as well, and in each neocortical column, they contain thousands of neurons, which are connected through synapses, synaptic connections. And the combination of these neurons and synapses, we can form ourselves a directed graph, which we can analyze with vertices and directed edges. So if we look at like one of these particular connections, we have two neurons that can be connected by a synapse, and this synapse is formed by axon and dendrites. And so what will happen is that in relation to this synapse, we have a presynaptic neuron and a postsynaptic neuron. And so the information can travel from the presynaptic neuron through to the postsynaptic neuron. And then as an analogy, to go out, we can just say like a neuron A in this instance is a source, and neuron B is our target. And this happens all through the connectome, so it's like fully connected. Okay, so we come back to the Bluebeam projects. They reconstructed many of these columns, and to be able to reconstruct them, they needed some data to be able to like perform these simulations, where each time like they performed simulations, they were refining the models further and further until it gave a likeness to a rat's cortical column. And on the right hand side, we see 6 sets here. And the first 5 of these sets are based on varying heights of the layers in the neocortex. And in particular, each set has different sets of morphological neuron types where they have different attributes. And the final set, the one colored in black, this is the set of average cortical columns or microconnectomes. And these just take averages of the first 5 sets we see here, the ones in red, blue, green, yellow, and pink. And in this final set, we want to look specifically at the final instantiation. So of these 42, we just want to look at the final microconnectome of the neocortical column, which we have here. So if we look at this microconnectome itself, this has 6 layers, and throughout the 6 layers we have 55 morphological neuron types, where again they have varying attributes, and between these morphological types we have excitatory and inhibitory neurons, where this is just one of the ways that they react with other neurons. And we also have approximately 31,000 of these neurons in this connectome, and they are connected by 8 million or so synaptic connections. We also take a look at how the layers are split in terms of their thicknesses. So in the deeper cortical layers, in layers 5 and 6, we have much thicker layers. So we have also more neurons here, more of the excitatory neurons. There's a larger portion of neurons that are excitatory, and they belong mostly in layers 5 and 6. And the upper cortical layers 1, 2, 3, and 4 are much thinner and just contain fewer neurons. On the image on the right hand side, we have the adjacency matrix for the connectome, so how like all the presynaptic neurons connect to the postsynaptic neurons. And we can see like a lot of the neurons connected through the main diagonal, sort of closely in relation to the self-contained layers. So for instance, like presynaptic neurons from layer 4 to postsynaptic neurons in layer 4 and things like this. And we have fewer interlayer connections, so we don't have so many connections in the off diagonals in here, the upper and lower triangles. So yeah, so we sort of see like a stronger levels of connection within the layers rather than between the layers. Again, so image on the left with some more statistics on the connectome. we wanted to compute the number of connections in each of these blocks in comparison to how many connections that there could potentially be. So for instance, with this first block here, the presynaptic layer 1 neurons to the postsynaptic layer 1 neurons, we have a 0.7% of connectivity in this block here, where we have, if we look on the image on the right, we have 848 connections between neurons in this block, and we have approximately 340 neurons in this block. So we just take the number of edges, which is 848, and we divide by the size of the matrix. So the number of neurons in this block squared. And by doing this, we find that the most densely populated blocks in terms of connections are between layers 4 and 5, where we see the highest levels of connectivity in these blocks. If we want to look at like absolute number of connections in each block, we see that the majority of connections occur in this final block here in layer 6, the presynaptic layer 6 neurons to the postsynaptic layer 6 neurons, where we have over a quarter of all connections held in this block here. So it's just a high level of complexity occurring here. Okay, so for our models, we want to have a few constraints. And one of these constraints is to split the connectome into blocks, where each block represents a layer-to-layer connection. So, I mean, for instance, this second block here represents the presynaptic neurons from layer 1 to the postsynaptic neurons to layers 2 and 3. And again, for instance, like this 14th block represents the presynaptic layer 4 neurons to the postsynaptic layer 5 neurons. This is one of our constraints. Okay, and the second constraint is a geometric one, where we wanted to compute the pairwise distances of all connected neurons in the BioM microconnectome. And once we computed all of these values, we wanted to bin them into bin sizes of 75 micrometers, which was the case for the general biological model. And the reason there was to be able to preserve soma distances of connected pairs of neurons in all submatrices, where the soma distances are just like this, the distance from the center of each neuron. So we will make reference to this distribution later on, in the sense like we want to use this as a sort of like probability distribution, where we want to take the largest value here, and we want to divide each column by this maximum value, so we get some sort of probability of connection when we measure the distances between neurons in our subsequent models. Okay, so we have 4 models we worked on: configuration model, geometric configuration model, block configuration model, and block geometric configuration model. And we use a cut, permute, rewire algorithm on all 4 models. On 2 of the models, we have a geometric constraint, and 2 of them we have a block constraint, which we discussed a moment ago. And we wanted to compare these to 2 models and the microconnectome. And so we wanted to compare them to the Erdos-Reyni model, whereby we wanted to randomly place a connection with equal probability throughout the entire connectome. So for the case of the Erdos-Reyni model, we just take statistics from the BioM microconnectome, whereby we have approximately 31,000 neurons. So if we look at the matrix that can represent this, we have almost 1 billion potential connections. And the BioM microconnectome has roughly 8 million or so actual connections. So what we did to compute the probability of a connection for each neuron pairing was to divide the 8 million or so connections from the BioM microconnectome by the size of the matrix, so like the 31,000 by 31,000 neurons. And this gave us a 0.8% chance of each neuron pairing to be connected, and making sure that we don't connect any neurons in the main diagonal, so we don't have any self-loops, because this is not what we want for these models. The general biological model, this one was to split the matrix representing the BioM microconnectome into 55 squared submatrices. And this is to represent each morphological neuron type. And within each of these submatrices, the pairwise neurons for potential connections were computed. And these were binned into bin sizes of 75 micrometers, which we suggested before. And within each of these bins, connections were randomly shuffled until we got some permutation of the original. And then we then what I did was to reassemble all of these submatrices once this permutation be completed, and this gave us the general biological model. And the BioM microconnectome is this is already what we've discussed beforehand. Okay, so the configuration model is our first model. We use the empirical data from the BioM microconnectome, and to be able to do this model, we represented the connectome in the form of a matrix of ones and zeros, where ones are connections and zeros are no connections. The rows are representing the presynaptic neurons, and the columns are the postsynaptic neurons. And so we read in the indexes of each connected pair. And so we get a list like we do in this first image, where we have the first neurons here, or like ordered here, and then the ordered pairs of postsynaptic neurons in this list. And to perform the cut, permute, rewire algorithm, what we did was to keep the postsynaptic neuron list static, and then we wanted to rearrange this presynaptic list in some form. So we get something like this list down here. And what we wanted to do each time was just to, for each neuron pairing, we wanted to make sure, like when we take a start point and we take an end point, we wanted to make sure that when these two are connected, that they are not the same vertex. And once we did that, once we did that for each neuron pairing, we had a new presynaptic list completed. So with this new presynaptic neuron list and the postsynaptic neuron list, we could reassemble our matrix then. And this gave us a representation of a directed random graph G sub C. Our second model, the geometric configuration model, it again uses the empirical data from the microconnectome, but with the added constraint of using distance dependence. So again, we take the presynaptic list as before and the postsynaptic list, and we keep the postsynaptic neuron list static, and we want to rearrange the presynaptic neuron list in some manner. So for instance, a first test is to make sure that the presynaptic and postsynaptic neurons are not the same. And then we want to make sure that it satisfies this distribution highlighted in blue on the right-hand side. So if we took our example in the center here, what we can say is the postsynaptic neuron is, say, the second vertex. And let's say, for instance, we chose the 8th vertex as a presynaptic neuron. The first check is to make sure that the 8th and the second neuron are not the same, which it just checks out. And the second check is to see if it will connect with some probability. And we can say, for argument's sake, that it's 50% chance of connecting. And in this instance, this 8th vertex doesn't connect. So we discard the 8th neuron to the presynaptic neuron list that we haven't used. And then we go again and we choose this time the 7th vertex. So again, we see that the 7th vertex and second are different. So it passes the first check of not being the same neuron. And then we see this time that it has possibly like a 90% chance of connecting. So in this case, we can just say, oh, yeah, it connects because there's a very high chance of connecting. So then we have a rewired pairing where we have the 7th neuron is the presynaptic neuron and the second neuron is the postsynaptic neuron. And we make sure to remove the 7 from the list so that we don't reuse it, with the ultimate goal of making sure that we have the same in and out degree for each neuron in the set. And once we've completed the new presynaptic list, and we can take the older postsynaptic neuron list, we can then re-index the matrix M, and this gives us a representation of a directly random graph G sub GC. And as a point of interest, when we recomputed the distances between these newly connected neurons, we found that we had an 84% mass in common regarding the distance distribution of the geometric configuration model and the BioM microconnectome, which is the highest mass in common that we have for all 4 of our models. Okay, so our third model is the block configuration model. This one again takes in the BioM microconnectome, but this time we split the connectome into these blocks before, where we just look at individual layer-to-layer connections. And so, I mean, in this case here, we just look at purely the presynaptic neurons in layer 1 to the postsynaptic neurons in layer 1. What we want to do in this block is to list out all the presynaptic neurons and all the postsynaptic neurons, and keep the postsynaptic neuron list static. And then we want to permute the presynaptic list in the same manner we did for the configuration model. And then once we've permuted it, we can then rewire these connections and just store these rewired, this rewired block. And then we just move through the blocks until we've completed all 25 of them. And then once we have all 25 blocks rewired, we can then put them all back together to reassemble the matrix M. And this gives us a representation of the graph G sub BT. On top of maintaining the same row sums and column sums representing the out degree and in degree of each neuron, we also maintain the number of connections in each block. So we have like 848 here, and again we have 848 here, which is it's supposed to happen this way since we're focusing on the blocks. The fourth model is our block geometric configuration model. This again uses the BioM microconnectome. and we split this whole thing into blocks again. But this time, once we split the connectome into these blocks, we want to compute the pairwise distances of each connected neuron set in the block itself. So we can refer to this probability distribution when we rewire each block. So we can see that by the fact that we are splitting this into blocks, and we're also computing the pairwise distances of each neuron pairing in the blocks, we can see that this is a combination of our second and third models, whereby from the second model we're taking that geometric constraint, and the third model we're taking that block constraint. And so we want to again take that postsynaptic neuron list, and we want to take the presynaptic neuron list, and we want to permute the presynaptic list in some way, just by utilizing these constraints that we've just mentioned. And once we've completed the first block here, so from presynaptic layer 1 to postsynaptic layer 1, we want to then focus on the next block and the next block and the next block. We do for all 25 blocks, and then once we've completed that for all these blocks, we can then reassemble the matrix M, and we get ourselves a representation of the directed random graph G sub BGC. Okay, so we can look at some results to be able to compare our models to see how much of a likeness they get to the BioM microconnectome. So our first metric is to use simplices, whereas simplex is just the generalization of like a triangle or a tetrahedron to some like arbitrary dimension. I mean, it's given this name since it's the simplest possible polytope that we can give in any space. Another name for these is a clique. So we have a few examples here. So 0 simplex is given by a single vertex. 1 simplex is 2 vertices connected by a directed edge. 2 simplex is 3 vertices given by 3 directed edges, where we have a source and a sink, and this is the case for the 1 simplex as well. And a 3 simplex, we have 4 edges connected by 6, 4 vertices connected by 6 edges, where we have again a source and a target. Okay. We wanted to compare the number of these simplices in all dimensions for each of our models. So we can see that the BioM model gives high levels of 2-dimensional, 3-dimensional simplices in particular. So we have like a lot of order in the connectome on the local level. We have high levels of complexity as well. So we have 4- and 5-dimensional cliques in the BioM microconnectome. So the model we wanted to compare against was the general biological model, which is given by this yellowy curve here. We find that the biggest differences between these models occurs in the second dimension. For our models in particular, we find that in each occasion, the models improve with their order. We have a large difference between the configuration model and the geometric configuration model, relatively speaking. And then we see a further increase in order here with our block models, where their values here essentially like overlap. because on some occasions the block configuration model gave better results than the block geometric configuration model in terms of absolute number of at least 2 and 3 dimensional simplices. And then sometimes the reverse happens, so the block geometric gave better results than the block model. But they both gave better results than the other models that we have created. So yeah, so having the block constraints kind of removed how can I say, the influence of the geometric constraint. So giving similar results here. A second metric we use for topological statistics are Betti numbers. These give a measure of the global connectivity of the connectome and the model. So to define them, so a 0th Betti number is the number of connected components. So here we have 6 vertices that are all connected to one another. So we have one connected component here. The first Betti number is the number of one-dimensional circular holes, and in our figure here we don't have any. So the first Betti number is 0, and the second Betti number is the number of holes or voids in our, at least in this figure. So we have these 2-dimensional cliques enclosing this space in the center of this object. So we have one void, let's say, in this figure. And as a further point, if we were to connect 2 vertices in this figure that aren't connected already, then the 2-dimensional cliques that are enclosing this void would no longer exist, and rather we would have sets of tetrahedra instead. There's another interesting thing where the Euler characteristic, when applied to polyhedra, has like a relationship with Betti numbers. So if we take our image figure here, we have 6 vertices, we have 12 edges and 8 faces. So when we take the alternating sum, we get 2. And similarly, if we take the alternating sum of the Betti numbers in each dimension, then we also get 2. And as a further point, we want to say the connectome that we have is fully connected. So as a 0th Betti number, we get a value of 1 there, since all vertices are connected in our connectome. Okay, so we wanted to compare Betti numbers between the models, and the highest dimension by which we could was the 3rd dimension. So we have here some non-zero Betti numbers for each of the 6 models. The BioM microconnectome wasn't computed for this because computationally it just wasn't possible. The first model here we have is the Erdos-Reyni model, where we don't necessarily see how many Betti numbers they have in the 3rd dimension, but in each realization we had anywhere between like 5 and 20. The next one I wanted to look at was the configuration and the geometric configuration model. We see like a large difference between these 2 models. And just showing how the geometric constraint influences the complex connectivity on a global scale between these 2 models. And then if we compare them to the block models, we see that a geometric constraint, when it's applied to smaller graphs, seems to have less of an impact on the order and complexity of the the models modeling the connectome. But we see that both having a block constraint and a geometric constraint, it had more order than the general biological model. And we also found that aside from the Erdos-Reyni model, that all 4 of our models and the general biological model had a non-zero fourth Betti number, and that in fact our block geometric configuration model had even a non-zero fifth Betti number. It goes in some way to showing the order and complexity of having these constraints applied to give us this model. So one of the things that was of interest for me, at least, was to state that computing these models, we used the same machine with the details, the basic details given below. We note that the configuration model, the block, and the block models gave very similar times. And this is mostly down to the fact that either they didn't have a geometric constraint, or they were just working on smaller sets, or both, especially in the case of the block configuration model. The geometric configuration model actually took a similar time to these other 3 models, until about 80% of all connections were made. It was just that the final 20% of connections that needed to be computed just increased the time exponentially. So, I mean, we found that because of this geometric constraints, a lot of the probabilities of connections became very small. So it just took a lot longer to actually finish computing this model. The Erdos-Reyni model, which is very quick to compute, since we're just placing a connection with some fixed random probability. And the general biological model, this took some time as well, since for each submatrix we had to do compute pairwise distances and then do some shuffling of the connections in a double for loop. So this computation became very expensive. Yeah. Okay, so just wanted to say that in terms of like complexity, our models had a similar level of complexity when compared to the general biological model on a local level, where we saw the biggest differences in simplices counts in the second and third dimension. And we found with some of these constraints that we were finding better and better likenesses towards the BioM microconnectome, but still a long way off, which is obviously showing a lot of order and complexity within this model. We found the biggest bigger differences between the Betti numbers when the geometric constraint was applied to larger graphs. Since if we were taking the Betti numbers on the block constraint models, the differences between having the geometric constraints and not having the geometric constraint, this reduced the difference in order and complexity of these models. And having the block and geometric constraint showed very high levels of global complexity when we apply it to the microconnectome. And if anyone wants to look at the how these models were constructed, we have a link here for you to look at. Thanks for listening. Any questions?
Yeah, please ask Kieran any questions directly. We have over 15 minutes left.
All right, could I ask a question?
Yeah, sure.
Yeah, I like the presentation. You know, the models that you constructed at the end when you were doing these Betty counts and or simplex count, do you do like on the average, or on just you constructed one particular model and then you counted the Betty? Or do you see, like, if you redid this if you redid this procedure, would these Betty counts change drastically?
No, so I maybe did like maybe I did like 50 like instances for each model.
Okay.
And so like kind of like took an average.
But what was the deviation between models? Because you know the thing is that the Betty numbers, okay, these Betty things tend to be very unstable. Okay.
Right. Yeah.
Right.
Yeah, I mean, so I mean, for instance, like at least with the fifth Betty number, I wanted to make sure like that I actually found this number, because I mean, you know, I couldn't do it just once and think like, oh, you know, I don't have a fifth Betty number, so I went over and over just to sort of double check like all these different values. And I was yeah, I mean, I was getting like values like these here fairly like consistently. So
all right, okay, all right, okay.
Yeah,
it's interesting. So that is also, you know, so generally Betty numbers are you know, it's not stable. Okay.
Yeah.
So that means that if you actually get some stability of them, that means that that's actually an information. That whatever moves you are making is makes the Betty preserve sort of these Betty numbers. So that's actually an information, you know.
No, I mean, I wanted to run this several times just to sort of like make sure like I had some results that were sort of came out sort of around some sort of value, and I just sort of like go for this value. I mean, like on the sort of like one of the more basic ones was the Erdos-Reyni, and each time we were having something like between 5 and 20 each time, no more, no less. So it's got like a little bit of a range there, and like for sure I understand that these are going to have bigger differences given like the counts of them. But yeah, I mean, these are sort of like sort of averages of where I was ending up with these.
I think Wojtek's question is really good, actually, because it might be fairly straightforward to put like a box and whisker plot on these things, right? So you have your 50 simulations, and you can give the range and the median.
Yeah, sure.
Yes, it's always too good to do that. You see, what did you use to calculate, to make this calculation? Did you use this Fluxter?
Yes. So there's like a Python binding.
Okay, all right. On top of Fluxter, I guess.
Yeah, exactly. Yeah.
Okay.
Yeah.
All right. Okay, good.
Yeah.
Very good.
Okay.
You know, another sort of, this is just a little warning, mathematical warning.
Yeah.
So, you know, the simplicial complexes and how to calculate Betty numbers and homology, this is really well understood and developed. Now, this is not the case for directed simplicial complexes.
Okay.
So actually, reality is that the paper for frontiers, right? There is like there's like a tiny paragraph about directed, but that is mathematically, it's, you know, this requires actually considerable reworking. So, you know, for undirected things, we know what Betty numbers is. For directed, suddenly, you know, we know what the simplicial complex is. People still cannot agree what is a directed simplicial complex.
Okay.
So community is like, because it's difficult, you know, like people come up with different definitions, and then if they typically come up with a definition that fits their purposes, and they prove something, and then you look at another paper that they use directed simplified complex, and they use entirely different definition that is not comparable to that. And they, so directed world of simplified complexes, it is not well established. Okay. So even these results, I mean, so there's always there's this one has to, I mean, so well, that's the only thing, just like little warning.
Sure.
I mean, this little paragraph in the frontier papers doesn't give justice to the state, to actually quite, I mean, this is difficult. That's the point. It's not that people cannot, it's just difficult. It's just difficult. And it is still in the progress of identifying even the definition, or like what is the directed simple shell complex, that, and you want to come up with a definition that reflects, understanding these directed networks or directed graphs, etc., better than. But that is still this is still a little grain. Of salt once treated. Very nice presentation.
Thank you.
Very cool. Very cool. Thank you.
And anyone else?
I have a question, actually. So this work, I should thank Wojtek and Martina. Wojtek is the topological data analysis group at KTH. So this project sort of came from a colloquium talk given by this researcher from Switzerland, and we should actually thank Svante Jansen for suggesting this geometric configuration model as a simpler alternative to the BioM model that we're comparing things to. Because somehow the question was whether just preserving the in and out degree as well as this layered information in blocks and the pairwise distances is enough, as opposed to what they were doing in the BioM model, which is way more complex. And the nice thing from these results, as far as I can see, Kieran, correct me if I'm wrong, is that you are able to get some of these petty numbers to be closer to, I mean, better than the BioM model, right? Up to definition of directedness. And the other thing is, in the link, so a lot of this is live research. The problem Kieran had was he was using his laptop to do all of this. We really need like 0.5 terabyte RAM to actually even compute the original model. So if anyone is interested, what can continue more exhaustive computations with this.
So sorry, I want to sort of, so I think this was really cool because, and in the original paper, this by Bluebeam, the Frontiers paper, right? There was this big difference, right, between these random models that they try to compare it and they are biological model. You actually got, halfway through to them. So that's a really good, idea. Like exactly, these networks have some additional constraints, and let's see, whether these constraints are enough to get. I think that's just great, super.
And
again, you maybe don't get all the way through, but I mean, definitely you are on the right track. So, we definitely need to look at the geometric constraints. We need to look at these block constraints. And then there is something else, right? So that's, so we just, that's progress, right? That's progress. That's, now we should look for something else.
Yes. I mean, I was going to say like our models like gave better results than the general biological model, which was the one that Michael Ryman did in this Fontaine's paper. But rather it didn't get quite as obviously good results like in the order and complexity as the BioM model. But I mean, it sort of got closer, like you say.
Exactly.
Another,
I have another question, because in that paper they really made they underlined that the number of these very high dimensional synthesis, 6 and 7, right? So somehow when you look at this actually paper, there is this even that for them that was somehow important. It sort of underlines that this doesn't happen. It's very hard to construct some models that has that property. For example, the existence of these high-dimensional simplices were not detected in the previous sort of just sort of stochastic models of these networks.
Yeah.
Okay.
Yeah.
Now, right, but you are taking there and you are modifying, right?
Yeah. So yeah, I mean, I'm taking like their BioM, the connections and their vertices, and just rearranging it in some way to obviously try and match like the complexity,
right,
of their models. I mean, like, yeah, I mean, so we don't have like as such high ordering as the BioM, and I mean,
but we keep up somehow. It doesn't destroy the number of of its high dimensional.
Yeah. I mean, like, obviously, like there's a lot fewer high dimensional simplices for the BioM microconnectome, but
but what I want to say, remember that in that paper, they really when I remember when they were writing and then it was published, one of the biggest thing from that paper was just the existence of high dimensional simplices.
Yes.
So, so the thing is that they actually had always the picture, like when they not this one that you are showing on a slide now, but they zoom on the end, and apparently that was not totally unexpected. That they're actually that they actually formed these even 6 or 7 clicks. That's like it. So what's interesting is that in your modification, I mean, okay, you didn't zoom on there. One would also ask a question: if I do these modifications, will the number of high-dimensional simplices change?
Okay, yes. I mean, I was actually working on trying to create these plots, and I think I ended up doing this, but like I just ended up losing like where I put the plots. But I did manage to zoom. I zoomed more in the 4th dimension because it just seemed like there just seemed like a bit of a range between the models there. But if we zoomed in on the, like, say, the 5th dimension, I mean, like, we wouldn't find all that many cliques like in our models, and I don't even believe like all of them even had 5-dimensional cliques. I think it's perhaps just the block models that had these 5-dimensional cliques, and the others, I don't believe they had them.
So what I want to say, let me just quote, like I'm reading now what they are, frontier paper, because I have it in front. Okay. Surprisingly, we found that reconstructions consistently contain directed simplices of dimension 6 or 7. This is the first indication of the existence of such a vast number of high-dimensional directed simplices in neocortical microcircuitry. And exactly. So somehow they really made it. They really made it. Like a big thing about this. So it would be interesting whether your modification changes, the number of 6 or 7 dimensional. And again, remember, those are much less, right? This is much less numbers. It's not 80 million. It's a much less. But apparently, just them being there and consistently being there. The question will be, when you produce your models, do you keep consistently, just having them, consistently, that you have them,
I believe like we got I mean, so at least for the 6-dimensional simplices, the only model that was able to sort of reproduce these was the block geometric configuration model. But it wasn't that they would consistently have these 6-dimensional simplices. It was, sometimes they did, sometimes they didn't exist.
Okay, all right, okay, that's interesting, because they got, I mean, again, like just comparing the numbers from 80 million, they really were happy that they were getting 18 7-dimensional cliques. 18, in this house, what? 18, and this, but that number apparently was consistent, like, there was like definitely always happening on 0. and they really made a big deal out of it. So the question is it biological? Like, do we really need this high order organization, or it is a consequence of their model?
I mean, right? I mean, like with since it's occurring in the block models, I guess there is like some like biological feature to this. I mean,
I don't know, like that's just like a generic question. I don't expect your answer, right? And just like a comment,
no, just
right?
like
Just sort of see. So again, like a 6-dimensional, they get consistently around. This is the average that they show: 4,100 6-dimensional and 18 7-dimensional. So it's like there's, and the question is it, does it reflect something interesting about this? Okay. Anyway, before that paper, that was apparently like no one even considered possibility of such.
But I think, Kieran, like it will be really nice to make the less than 10 million, because it's a scale of 1 × 10⁷, right? Maybe the lower numbers, you can zoom in and show this on the sort of maybe log scale, log-based 10 scale, and also put the box whisker plot.
Exactly. It will be sort of interesting to see the box. Exactly.
Yeah, exactly. Because I think, also Svante would be interested in this, because our basic intuition for the seminar the colloquium was that block geometric configuration should, at least it'll be nice to know what's captured by preserving the layer structure and the within layer and in between layer
the distances.
Yeah.
So in that sense, it might be nice to see, especially in the higher dimension on the right scale, it'll be nice to see what the distribution is.
No, I mean, I like I said, I got at least I got like a zoom in box version somewhere. Unfortunately, I just didn't have it for this for the slides.
No, good.
But yeah, there was.
Are there any other questions? Any questions from the students? There are several master's students here. Okay, if there are no other questions, let's thank Kieran for a great presentation and a lot of work. And thanks a lot. See you all later.
Thank you, Kieran.
Thank you.
Bye, Raz.
Bye.
That was great.